From Drexel University's School of Biomedical Engineering, we present the cell culture bioreactor with novel mixing and loading. Cell-based therapies are a recent technological expansion in tissue engineering. However, they are still not often used in clinical applications. Bioreactors have been used to attempt to bridge the gap, but current solutions are inadequate. Of the two existing solutions, they both only fulfill one of the three criteria that we considered. Based on this need for a robust and flexible bioreactor system, our system features mixing and dynamic loading capabilities.
We considered some requirements to target when prototyping and constructing the final product, including cell plates, dimensions, required force resistance, mixing capability, and the system's durability. Due to the COVID-19 pandemic, however, we could only test 2 out of the 4 requirements before the lab shut down. The choice of mechanical ports was also influenced by our budget of $500 granted by the School of Biomedical Engineering. Bioreactors with a loading mechanisms have been found to use load cells, which can be costly and way out of the budget. Nevertheless, our approach is novel, such that we use a force loading approach via a pulley system to provide a different way to measure the stimulus on the sample.
Our 3D model presents our system featuring some close-ups on the lid and plate. This lid and plate combination features unique structures to promote mixing and therefore the exhaustion of medium nutrients. The cell plate, currently suited to hold a maximum of 24 individual hydrogel scaffold samples, features an inset within the well. Samples are intended to sit within this inset to prevent excessive lateral movement during incubation. The lid has 24 complementary pillars with geometry to reflect the empty space in the well. When the lid is lowered into the plate, the lid will shift the medium such that it creates a displacement in the medium, raising the fluid level and promoting mixing due to convection. To verify our device, we performed a release study to observe the differences in the diffusion coefficient between a static, mixing, and loading condition. To calculate the diffusion coefficient, we created a computational model using an approximation from Ricker and Pepys of fixed first law. Our initial results found mixing to be as effective as loading. As such, samples that do not require a mechanical stimulus will also experience similar benefits relating to mass transports. These benefits include effective movement of nutrients in and waste products out of the scaffold. Additional findings were unable to be attained due to the COVID-19 pandemic.
This is our most recent prototype of our bioreactor, made in 6 months during the academic year. The cage structure is made of aluminum and various stainless steel parts, mostly from McMaster car or home improvement retailers such as Lowe's or Home Depot. You can see our 3D printed lid and plate, the lid controlled by the pulley system. These parts were printed with PLA during design and prototyping for faster turnaround, but ideally would be made of less quickly degrading material for users. Our pulleys are made of stainless steel, and our nylon thread is controlled by our NIBIB 17 stepper motor. To conclude, this bioreactor was created with the intent to contribute to expanding opportunities for personalized cell-based therapies. This would bridge the gap between currently researched solutions with a clinical application for patients who seek alternatives to their treatments. We would like to thank and acknowledge our advisor, Dr. Spiller, for guiding us through the project, as well as everyone else who has helped us with instruction or experimental support, and thank you for watching.